Okay, the cell cycle, including mitosis and meiosis. So before we start each processing, we are going to go through the general concept. So your body cells can be divided in two categories. Number one, body cells, also called somatic cells. Number two is your sexual cells, also called gametes. So here, I want to particularly address that your gametes must mate in special organ. Otherwise, is no good. You have disease. Okay. So this is your chromosome. Okay. So normally, if you have 46 chromosomes, you have 23 pairs. So you have pair 1 and pair 23. And pair 1 to pair 22 is containing same genes, whereas 23 depends. If you are female, then you have two X chromosomes, so they actually containing same genes. Well, if you are a boy, then you have X and Y, so then the two completely different chromosomes, they containing different genes. So this is your chromosomes pictures before they divide or after they divide. So in other words, if your cells are not dividing, this is your chromosomes picture. If you are preparing for dividing, then you have to duplicate your DNA. So this is your finished interface. So you can see each chromosome has two chromateins. So I make this bigger. So you have two chromateins per chromosome. You have centromere linked. Okay, so now in here, you can see here, right? So you can see here because this chromateins are two chromateins are identical twins. Means that they're exactly the same because you use one copy to making another copy. So in the human, your chromosomes is linked to histone protein. So you must dissociate your histone protein to do DNA replication or transcription. So now, let's talk about another two concepts. Diploid cells means that your cells containing 23 pair of chromosomes, 46 chromosomes. So this is non-dividing cells. This is after interphase, they are going to divide. Okay, most of the time, your cells is non-dividing cells. So this is called diploid cells because you have D means double, you have pair. Whereas here it's called haploid cells, means only half amount of chromosomes. So this is why you actually finish meiosis. You only have 23 chromosomes. So for the sexual cells, you can have X or you can have Y. You cannot have both. And this is between the meiosis 1 and the meiosis 2. However, you only have half amount of chromosomes. So this is called haploid. So this is a picture of your mitosis. So you have to do preparation. So this preparation called the interphase. So they containing G1 as G2. So this is G0. So you can see in G0, And G1, you get this diploid cells. And you have a two of each pass. So you have one from your mom, another one from your dad. Then S phase, so when you go to G1, you have to check whether your cells are normal. The cell contents, the nutrients, the DNAs. If anything abnormal, you kick out. You are not eligible to go this interface. So then if everything is all right, then you go to S. So you duplicate your COM-Ts. So now each chromosome has two COM-Ts. Okay, so two sister COM-Ts should be identical because you're making a new one copy from another one. Then you go to G2, you have to double check DNA, the contents, the cell size. If everything normal, you go ahead for mitosis. Okay, so now let's emphasize, you must duplicate your DNA, otherwise you cannot making identical cells. So here, you please replicate DNA before you vote for mitosis, okay? So after you do replication in S phase, you have two COM-Ts per chromosome. Okay, this is a picture to show you here. So you can see here. So initially, you only have 4 COM-Ts, right? And 4 chromosomes, because each chromosome containing only 1 COM-T. Then you replicate, then you become 2 COM-Ts per chromosome, but still 4 chromosomes. Then you divide, you're making 2 identical daughter cells. So these 2 are identical, and those 2 also identical to the parents. You only divide once. Okay, so here occur in somatic cells, your body cells, you produce identical cells, number of divisions only 1, you making 2 cells. Okay, so it's a diploid cells become diploid cells. Okay, now let's have a look. So this is a pictures of onion root. So that's the part, right? So let's start with your mitosis. So mitosis start with here, division prephase. So what you can see is here is prephase. So you can see the nuclear orders disappeared and nuclear become condensed. That's why you can see this. And the chromosome become visible. Okay, so then you have 3 changes for your nucleus. Number 1 is the nuclear orders changed. The nuclear orders disappeared. Number 2, this chromosome become very condensed, and also nuclear envelope broken down. Okay, so that's 3 changes in the nucleus. You also have 2 changes in the cytosol. So you can see your centrals linked to spindle fiber, and then they move to 2 ends. Okay, that's prephase. So let's go for metaphase. So metaphase is here. So you can see here. So this is a picture for this. So what's happened? So you can see because these centrals and the spindle fiber, they pull the chromosomes on both sides. So that's why they lie on the middle. They lie in the middle plane. So you can see this spindle fiber touch to both sides. Okay, so let's look at detail here. So this is a centromere, but the outside called keritochore. Keritochore is linked to spindle fiber. The spindle fiber linked to your chromosomes. Okay, so now let's go ahead. So anaphase, you can see that your both sides. Okay, just wait a moment. So why I got so many students? I got 2 Michaels. Okay, good. So now let's continue. Okay, so you have and this one. So you can see the sister chromates separate, right? So what you can see here, the 2 separate. So therefore, you got 8 chromosomes. You got 8 chromosomes here. Because when they separate, they become chromosomes. So you start with 4 chromosomes, now it's 8 chromosomes in anaphase. Each side you have 4. Okay, so you can see which one? This one. This one is anaphase. So finally, we go to telophase. So telophase really is considered 2 cells now. Only this small part linked. So you can see this new cells has a new nucleolus and the new envelope, right? New envelope. And also your chromosomes become less condensed because they actually dissociate with your histone protein. They need to making their own mRNA to making protein for the new synthesized cells. Okay, so now let's call telophase. Okay, now, so that's an illustration of your mitosis. So number 1, your dad gave you 1, your mom gave you 1 to form your body cells. Then interphase, you actually duplicate your DNA. So you have 2 chromates per chromosome. Then during the mitosis, the 2 chromates separate. So you're making 2 identical cells. Okay, so now you have to completely separate the 2 cells. So let's have a look. So for animal cells, okay, so you use a microfilament. We already mentioned this one. You microfilament form a ring between 2 cells. Then they cut from periphery to the center. Okay, so that's the one. So you form a cleavage forum because they cut from periphery to the center. Whereas plant cells, you are not using these mechanisms. You use Golgi body, so you have so many vesicles. So they lie in the middle and form the cell plate. So then they cut. So they cut from center to periphery. Then you don't need from cleavage forum. So let's put everything together. So here, recap. Okay, so now you're doing interphase, you have to go interphase. Then if everything is okay, then you go to mitosis. Okay, so mitosis is come with P, M, A, T. Then finally you have cytokinesis become a 2 identical cells. So then one most of cells will back to G0, and if you still need to divide, then they can enter to the cycle divide again if you cut your fingers, for instance. So certainly all of your body cells should have potential to divide, but be careful though. Some cells has a high potential, such like your skin cells, your stomach cells, your liver cells. Some cells has a very low potential, such like your brain cells, your nerve cells, your muscle cells. So if you damage your brain or you damage muscle, almost no chance to back completely. Okay, so that's we talk about mitosis. So normally that's what happened to help you grow, to replace your old cells. But you do have abnormal mitosis. That's cancer. Okay, the cancer is also considered mitosis, but is abnormal mitosis. So let's summarize. So you're making identical cells, the identical each other, and also identical to the parenting cells. So the purpose is to maintain the chromosome number from generation to generation, right? So now let's try to memorize this name. So prephase, P for preparation, metaphase, M for meat, anaphase, A for part, telophase, T for tear. Okay. So now let's try to do this one. So one thing you probably forget is what's MTOC. So MTOC is called microtubule organized center. So when you have that, it's in prephase. So prephase on here. So this is called this part central, including the spindle fiber called microtubule organized center. Okay, good. So now let's back to the question now. Okay, we have problem, right? We have problem with number one. This is really review and the cell structure, right? So what's the concept here? So the concept is only plant cells has central. So plant cells, sorry, only animal cells has central. Plant cells do not have central. Plant cells do not have central. Plant cells do not have lysosome. Remember that? Okay, so now the answer for this one is C, because plant cells do not have central. Okay. So now let's look at this one. So question 2 is a B, question 3 is A, question 4 is C, question 5 is D. Yeah, you did very well. Okay, don't forget question 1, please. Okay, so now let's answer this question now. Each cells, okay? We talk about each cell. Each cell. So you start with 12, end with 12, correct? For each cells. Okay, you do need to practice because when I talk about it, it seems to be easy, but when you practice, it's very easy to make a mistake. Okay, so I have a lot of questions for you to do practice. Okay, so now let's talk about meiosis. So what's meiosis? Making germ cells, right? So remember, your germ cells are haploid cells, only containing half amount of chromosomes. So in the meiosis is you're making your sexual cell. So you go through 2 time division. You only replicate DNA once. So that's why you're making 4 cells. They are haploid cells, only have half amount of chromosomes. For instance, you start with 46, you end with 23. And then also there are different types of different cells. They are unique cells, Okay, so let's go through this one. So number 1, they have to happen in a certain organ. So for human, you get ovary to making egg. You get testes to making sperm. If they mate in any other place, then it's wrong. So number 1, they have to do replication. They have to go to interphase. Okay, remember, any division have to go to interphase because you have to check, you have to replicate your DNA. So here, same thing, you have to replicate your DNA. You can see you start with 46 chromosomes, you duplicate your DNA, become 46 chromosomes, but each chromosome has 2 identical quantities, Okay, so that's the whole picture. So again, you have to do interphase. The interphase is must. Then you go to meiosis 1. Then you go to meiosis 2. Then you're making 4 individual cells, 4 unique cells. So the terminal cells, you cannot back to this cycle again. Okay, so now let's start. Let's start with meiosis 1, preface 1. So in mitosis, we said you have 3 things change in the nucleus. Number 1, your nucleolus disappeared. Number 2, your chromosome is become very condensed. And number 3 is your envelope broken down. They also happened in the meiosis 1, preface 1. But they have additional change. You have to pay attention. It's crossover. So what do you mean by crossover? So you can see on the left side is a parental. On the right side is maternal. So your father and your dad's home team exchange, they call it crossover. So the touch area is called the synapses. So hemlocks means that your mom's one, your dad's one, they called hemlock chromosomes a pair. They are highly similar, but they are not identical. Okay, so hemlock chromosomes pair with each other. Then you can see you got exchange. So this exchange point for chasmata. And this area, so your mom's one containing your dad's one containing your mom's one. So that's the first time they become different, right? So this can occur anytime, any place, any place. Okay, good. Crossover. So here, what you can see is this is centromer. So this is two crossovers. That's why I said it can happen in any location. So this is electron microscope. So second one is for the microphase one. So what you can see is here, they lie in the middle, right? Lie in the middle. So this is mitosis, this is meiosis. So what's the difference? Although they got this one here, so they actually got this microtubule organized center. They actually touch to chromosomes, but be careful. The chromosomes only touch to one side, not both sides. That's quite different from your mitosis, right? So now you can see that's assume that this magenta color is your mom's one and the blue color is your dad's one. So they actually pair each other instead of lying in the middle. So the pair, but of course you can go to left, you also can go to right, it's your choice. So this is called hemlock chromosomes pairs, the pair. So anaphase, you can see here, so the pair separate. The pair separate means that your mother's one and separate from your dad's one. So the bottom picture is mitosis anaphase. That's quite different, right? Because here, what is that? It's Comte's exchange. So left equal to right. They are identical. This time it's not because left side is your mom's and the right side is dad. And the second pair, left side is your dad, and right side is your mom. So you free combine. Okay. So now, after this, they become a haploid cell because you only have half. So you start with 4. Now you only get a half for each side. Okay, so they are haploid cells. They are haploid now. So this is telophase. So you can see there are 2 newly synthesized cells, but each side you only have 2 chromosomes, reduced to half. Okay, so you can separate your cells or you can leave it until later. So let's go ahead for meiosis 2. So meiosis 2, you actually go straight from meiosis 1, no replication and so on. So what you can see here is, you can see, right? So this one, well, if you are not going to divide, then they can go to mitosis 2, right? So you can see here, so these chromosomes were separate. The chromosomes were separate, right? So the chromosomes were separate. So here in the meiosis 2, you got Comte separate. But unfortunately, these 2 Comtes are not identical because you already done crossover. So then after this, you actually making different cells. You're making 4 different cells. So this is again mitosis anaphase. This is anaphase of meiosis 2. So what's the difference there? because your quantities are no longer identical because of those crossover. Okay, so now, so this picture, see this one, although they actually separate the meiosis one, you separate the fathers and the mothers, but the meiosis two, you separate chromates. But unfortunately, these chromates, these two chromates are not identical, okay? So you're making 4 different cells, and each cell only have one set chromosomes, only have 23 chromosomes. Okay, so now we can understand this picture much better, right? So you interface once, so means you duplicate your DNA only once, but you divide it twice. And then you're making 4 different cells. Okay, so you can see how the chromosome change. So this is when they divide once, so they become haploid cell. Then they divide second time, still haploid cell. Okay, so that's the picture illustration. Okay, so what you can see is this above one is meiosis one. So you actually have to remember that because crossover in the prephase, because of this called anaphase, they actually separate randomly. So those two were making not identical cells, different cells. So you can see the above one is different from the bottom one. Above one is got 2 chromosomes from your mom, 1 from your dad. Whereas bottom one, you got 2 from your dad, 1 from your mom. They are not identical. Then you can divide again in meiosis 2, although you separate chromates. But because the chromates are not identical anymore, so then you're making 4 unique cells. They are haploid cells. So let's put mitosis and meiosis side by side. So mitosis is making identical cells. And you only divide once. You have diploid chromosomes. You still have both your parenting chromosomes. Whereas meiosis, okay, they end with 4 different cells. They are not identical. What's in common for those 2 divisions? Interface. Interface is the same processing. So you have to check G1, you have to go to S, replicate your DNA, you have to go to G2, you have to check again. Okay, good. So this is a picture to help you to do revision, okay? So you can do this later. So I also make a table for you to do revision. So let's go to meiosis. So meiosis only occur in the germ cells. They produce your sexual cells. You got 2 divisions, meiosis 1 and 2. You're making 4 unique cells. So the cells is haploid, only half amount of chromosomes. Crossover occur, and also hemlock chromosomes were separated in anaphase 1, but the 3 combined. So that's why you're making 4 different cells. Okay, so now let's give you a very interesting question. Okay, so this is very interesting and a very challenge. Be careful, okay? I guess only probably 1% or 2% student can make it right. So let's see. Can you make it right? Okay, do it, please. So you just type T for true or false with F. Okay, let's check. Let's check. Okay, so look at the picture. Okay, well, so force, choose force. What do you think? Do you know why? The answer is FTTF. So you know why? See this one? So this is a picture. Okay, this is picture. So you can see 1, 2, 3, 4. So they ask you whether it's anaphase of mitosis or is anaphase of meiosis. So how can you see the difference? So see this one? So this is anaphase mitosis. So you get 4 chromosomes. So those 4 chromosomes are identical, right? From this side. That's number 1. So number 2 is you get 2 pairs. So each one you get 2 pairs. So the red one is the same as the second one. So the pair, sorry, not same. So this is first pair, this is second pair, right? So here, this is your mitosis. How about meiosis? Anaphase 2. So you don't have pair anymore, right? Because your primary chromosome is already separate. So each one are different. So look at those 2 pictures, then look at these question pictures. Those look at this one or this one here. Which one they look like? It's more like this one, correct? Because all these are different. So each side, look at this side. So the 4 different chromosomes. So it's more like this one, not that one. Okay? So you look at this one. I'll give you 30 seconds to read. Okay, done. So now let's go for the fundamental question. Okay, so now let's do this one. Hopefully you won't make any mistake, right? We'll write it down all of this. Okay, answer for 13. Hey, good, very good. Half, right? Half mana chromosome. 14 is C, 15 is D, and 16 is E, and 17 is B. Perfect. Okay, very good. Okay, so let's look at this picture. So your whole life can divide into division. So start with mitosis. So what is the pink area? Mitosis. When you start mitosis, since you are one cell, since your mom gave you egg and your dad gave you sperm, they combine, become zygote. So the fertilized egg. So now, since then, you start with mitosis. So this is called embryo development. Then when you mature enough, then you can make in your sperm or egg, okay? So now, let's have a look, right? So this becomes cycle because you have this fertilization, okay? But this pink area never stop until someone died. But this one, meiosis has a timing, right? Because you have to reach the puberty to produce a sexual cell. So we'll talk about this more later, okay? So this you have to know. Okay, so now let's talk about the genetic variation. So we actually say everyone are different. Why you are different? So someone say, oh, mutation. Okay, mutation could have, but it's not a major contributing factor. So what's the major contribution factors? 3, okay, remember, meiosis, crossover in prephase, independent assortment of hemlots in the meiosis 1 anaphase because they free combine and separate. Okay, so theoretically, none of the sex cells are identical because you can randomly separate. And also, join the male and female gametes, the fertilization is 100% random, unpredictable. So these 3 are major contribution for your genetic variation. So someone confused with number 2. So let's talk about this number 2, okay? Independent assortment of hemlots. So look at this one. So let's start with you have 4 chromosomes. You only have 2 pairs chromosomes. So then you can have these 4 different combinations when you finish meiosis 1, because you can have 2 is from your dad and 2 from your mom, and 1 is from your mom, the small one, the big one is from your dad. Or you can have a big one from your mom, the second one from your dad. So you have 4 combinations, right? So this is, you actually only have 2 pair of chromosomes. So we have 23 pair of chromosomes. So imagine how many different choices you have. Very interesting, right? Okay, 3 major contributions for your genetic variation are crossover, independent assortment of hemlots, chromosomes, and fertilization. Okay, so now let's talk about your regulation. So your cells only divide when your body needs to. We just talked about it yesterday about the energy. Same thing happened in the cell division, cell division. So what's the checkpoint? Okay, checkpoint. So number 1 is a checkpoint. checkpoints, G1 checkpoint, you have to check cell size, nutrients, growth factors, DNA damage. Okay, so you have G2 checkpoint. You check the size, DNA damage. You also have here, so during the meiosis or mitosis, you also have another checkpoint. So when they touch to when you chromosome touch the spindle fiber, they have to check. You have to touch equally. So you have 3 checkpoints. So this is controlled by cyclin-dependent kinase, the enzyme and substrate set. So number 2 is cell size limited growth. So we actually say you have a surface area which they can cope your value. So if your cell is too big, then they cannot cope because we already spoke about your memory transport. You have to get in nutrients, you have to get in water, you have to get rid of waste. If you don't have enough the surface area, you cannot do it, especially if your cells are too big. So this is examples, just give you simple examples. So this is surface area, right? So let's assume that is a Corvette. So the size is 1 millimeter. So then the surface is 6. The value is 1. So if you have a 10 side, 10 millimeters, then your surface area is 600. Then your value is 1000. So you can see this dropped dramatically, correct? So you can see this is ratio is 6 to 1, or this one is a 6 to 10. Okay, so this is dropped too much. So if your cells is too big, then your surface area is not enough. So you're not good. Okay, not grow too big. And also your genome is fixed. So if your cells are too big, it means that it requires a lot of protein. Means the wartime, the making mRNA is no good. And you're making your DNA overload. Okay, others is also contribute, but those two is frequent happened in your past questions. Okay. Okay, so this is we just said here. So cyclin-dependent kinase are the controller enzyme catalytic reaction, right? So this is how they control. Okay, so I have some notes you can review, you can use to review. So now let's talk about abnormal growth. Okay, so we actually say you have to grow, no problem. Come on, like you, very young, grow every day. And for me, I'm older. I probably do not grow so fast, but I do need cell division. Why? Because, come on, I have some dead cells need to be replaced. Okay, so our cells, our body need grow. We also need to kill some unuseful cells or bad cells. This is called apoptosis, also called programming cell death. Okay, so you may wonder, when do I need that? Okay, during the cell division. So here, if you actually grow up normally and your cells has a lot of this called here, tumor suppressing genes. So they will stop you, not grow, okay? So if you don't have those genes or some genes are mutated, then the chance for animal, they can have a tumor. Okay, just give you examples. So this one, P53 are very famous protein. Why? Because they are tumor suppressing factors. So they can stop tumor. And also they have a lot of partners. So this is one kind. So they can influence P53 functions. So let's work out what's the p53 functions. So you can see here. So if you grow normal, that's okay. You don't need p53. If you are not, they will stop your growth. So you have to repair your DNA. If you are repairable, you can restart. You can enter to the cycle again. Otherwise, they will ask you to kill yourself, apoptosis. So from these two ways, they can control that you have normal cell division. So then, if they're active, means that they can control that. If you add another factors, they may actually regulate the p53, so that you can modify them. Okay, good. So now, let's do some more questions. So look at this one. Look at this one. When you start to do mitosis, when you start to do meiosis, okay? So let's start. Okay, so let's go through together, shall we? So let's start with number one. Okay, good. Let's start number two. A fertilized egg here, correct? It's already been fertilized. So then it's also A. So I answer, someone didn't answer, you panic. So it's here, here, you already combine two germ cells. So you start with embryo development, still A, mitosis. 3B, no problem, right? So 4 is A. 4 is also A because the ovum is also another name for egg, fertilized, already fertilized. So 5 is A, easy, and 6 is A, also easy, right? Abnormal growth. And 7 is B. Good, okay, perfect. So let's go ahead. So let's do more questions. I think this probably is too simple for you. So let's have some little bit challenging question. Okay, sorry about that. So let's try this one. This one seems more interesting and challenge. So now let's put this one. So these pictures for these two questions, and the bottom pictures is for 3 and 4. So 1B, 2C, 3C, and 4C. Yeah, good. So I don't need to explain any more than. So now let's try even more challenging question, okay? Okay, so we have problem with 9 and 10. So I don't think you have problem with 11 and 14. If you do, you have to do revision, okay? So let's start with this one. So this is really a key concept, okay? So let's look at this A. So you can see you have 1 pair, 2 pair, 3 pair, and 4 pair. So you have pairs, right? So if you have pairs, means that that's body cells. That's your body cell. That's a somatic cell. Okay, so you have 4 pairs. So how about this one? You have 2 pairs, right? 1 and 2. So there's still body cells. So how about this one? So you don't have pair. You have 4 chromosomes, but they're all different. So this must be your germ cell, correct? So for this one, so each one, you have 2 pairs. That's wrong. Because each one has 4. They shouldn't have 4. They only have 2. Because your mom gave you 1, your dad gave you 1. So this is wrong. So number 1, you cross this one out. So they were not correct because you never had this one if you are normal. Okay, so this is abnormal. So now let's see this one. So let's read this question. Okay, so 2n equal to a means that each n is 4. Okay, you have 2n. So then, do you have n, 2n? You don't have 2n, right? Okay, you don't have 2n for this one. Okay, you do have 2n, but unfortunately, you don't have n is not equal to 4 because n is equal to 2. 1 and 2. So this one also wrong. Okay. So then the answer for this one for 9 is A, correct? You only have one choice because you 2 are equal to 8. So let's go to C. A gamete. So the gamete must be this one, C, correct? Okay, so now let's give you answer that. Sorry. Okay, so for 9 is A, 10 is C, 11 is C, and 12 is B, and 13 is A, and 14 is D. Okay, so now let's get more practice, okay? So let's try to do this one, okay? This one. Oh, I give answer already. Okay, that's good. Now might I give you another set of questions, okay? So if I gave you answer already, okay. So now let's give this one then. Okay, good. I think you got right answer, right? So okay, so let's talk about a little bit about this ratio of value to cell surface. Why is so important? Okay, so now let's give this picture. So you did right, so I don't need to worry about your answer. But I do want to show this one because this can be very difficult question. If you don't have a general concept, you may miss out. Okay, now, okay, so this is here. So we said this one. So let's say if you, the COVID, right? So here, so you have two, that's each side is two. So then what you do is 2 multiplied by 2. So let's do 1, okay? Let's do the 2. Okay, so this one for the surface area. So surface area is what? Is, let's try this one. So it's 2 multiplied by 2, correct? Yeah. And multiplied by 6 because you have six area, right? So that's how you get 24, correct? This one. So it's a side and multiply side and the 6 side. So how about the value? So let's do value for this one. So you get 2 multiplied by 2 and then multiplied by 2. So side, side, and side multiply. Okay, so now you can see the ratio, surface area ratio, surface area, and the value ratio is 3 because 24 divided by 8 is equal to 3. So then you get 24 divided by 8 equal to 3. No problem, right? So now let's move to this side. So move to this side, correct? So just do this one. The third one is much simpler. So again, your side is 6 multiplied by 6 multiplied by side 6. So you got 216, correct? Okay, so now what's the next one? Your value. Your value is the same because 6 multiplied by 6 multiplied by 6 also equal to 216, right? Then what you have is 216 and divided by 216, right? Because this divided by this equal to 1. What do you think? So you can see the ratio, surface area and the value ratio decrease dramatically. So your cells, if it's too big, like this one, like this third one, you won't have enough surface area to get in the nutrients and the gas and get rid of the waste. Okay, so the answer for this one is C. Great. Okay, so I'll give you a break. Now we can come back. Okay. So heredity, genetics. So the genetics can be divided in two periods. Okay, so the old period is Mendelian genetics, the father genetics. So the three laws are fundamental for the nowadays genetics. So now let's start with a general concept. So at the beginning, we must review what's the allele and the gene, what's the homozygous, what's the heterozygous, genotype, phenotypes, dominant and recessive. Okay, so let's watch these short videos. So you have to learn that. Okay, so someone say you have to understand that actually it's memorization. So you have to memorize. So let's start with this video, okay?
You should then be able to describe what's meant by homozygous, heterozygous, genotype, and phenotype. Now, this video does contain a lot of definitions, and it's really important that you learn them. I would suggest that you watch this video several times until you're happy with the ideas. In previous videos, we've seen that DNA is found in chromosomes, and I'm showing you here a human chromosome. Remember that normal human cells contain 23 pairs of chromosomes. So that means that normal human cells contain 2 copies of every chromosome. And I'm showing you that here. Now, a key fact that you need to learn is that one chromosome in the pair comes from your father, and the other chromosome comes from your mother. We also saw that genes are sections of DNA on a chromosome. And again, because chromosomes come in pairs, we've got 2 copies of every gene. I'm showing you here the gene which controls earwax. We're looking at the earwax gene because earwax is controlled by a single gene. Most characteristics are controlled by many genes acting together. Now, genes often come in different versions, and scientists call these alleles. So alleles are versions of a gene, and that's a key definition that you need to learn. The gene for earwax has 2 alleles. One allele leads to wet earwax, and we're going to give this allele the symbol capital E. The other allele leads to dry earwax, and we're going to give this allele the symbol lowercase e. As we said before, we've got 2 copies of every gene. So imagine that a person has 2 copies of the allele for dry earwax. I'm showing that here. The genotype of a person tells us the alleles present. And again, that's a definition you need to learn. So this person's genotype is lowercase e, lowercase e. Because this person has 2 copies of the same allele, scientists say they are homozygous. Okay, now the phenotype of a person tells us the characteristics caused by the person's alleles. So because this person has 2 alleles for dry earwax, their phenotype is dry earwax. Okay, this person has 2 alleles for wet earwax, so their genotype is capital E, capital E. And again, because both alleles are the same, they're homozygous. So because this person has 2 alleles for wet earwax, their phenotype is wet earwax. Okay, here's a third person. Their genotype is capital E, lowercase e. In other words, they've got 1 allele for wet earwax and 1 allele for dry earwax. Now in this case, the person has 2 different alleles. Scientists call 2 different alleles heterozygous. So this person is heterozygous for the earwax alleles. So what phenotype will this person have? Well, in the case of earwax, the allele for wet earwax is dominant to the allele for dry earwax. A dominant allele will show in the phenotype even if there's only one copy present. So in this case, because the allele for wet earwax is dominant, this person's phenotype is wet earwax. The allele for dry earwax is recessive. A recessive allele will only show in the phenotype if two copies are present. In other words, if no dominant allele is present. As we saw before, this person has two copies of the allele for dry earwax. So because there's no dominant allele present, their phenotype is dry earwax. Now, as we said at the start, some characteristics are controlled by a single gene. However, most characteristics are the result of many genes acting together, for example, height. In the next video, we're going to look at genetic crosses and see how alleles are inherited.
So in this video, they talk about a dry wax, and the wet wax. Okay. So now use these samples to illustrate this concept. So what's allele? Alleles in this video is a big E and a small e, correct? So big E and small e. So what's a gene then? So the gene means that the code for particular protein. So the alleles is only one version or two versions of genes. Okay, so the genes in this video, they said, is the wax. So the alleles means a different version, dry version and white version. So let's see the homozygous. Homozygous in this video, they could be double E. So let's try to do that. Okay, so then we say homozygous in this video is all correct, right? No problem. So the heterozygous for this one, genotype for this wax, you have three types, right? So you have three types. So combination of alleles. So what's this one? Phenotypes. Phenotypes, you only can have two. The one is dry or wet. Because the expression, they show up. So who is dominant in this video? Big E, right? Big E is wet. And which one is recessive? Small e. Small e is dry. Okay. So if you still have problem with this concept, please watch the video by yourself. Okay. So now, let's review the further genetics 3 laws. Okay, they are fundamental for nowadays genetics. You must learn them. So one thing we have to say at the beginning is, at that time, the father genetics does not know the genes. So he did not know. So then he used trees. So now in this class, we use alleles and the genes. Okay, so now let's start with this one. So here is a law of dominant. So here, so you can see one parent is big, another parent is small. When they cross these two parents and they get order of the offspring is tall, very tall. So then they actually have this hypothesis that here, this big one must be dominant, then the small one must be recessive. So then this is the Punnett square. So Punnett square are absolutely 100% essential for you doing genetics questions. Okay. So some students say, oh, my brain is good. My brain is good too. I always have a good brain. But for past more than 30 years, I use Punnett square because this can make sure that you get 100% correct for your question. Okay, Punnett square. So how you do that? Easy. So here, your father is a big T, big T. They are homozygous, correct? Your mother is small t, then they also homozygous. So you cause them, you can have big T, small t for the children. So because the big T are dominant, so they show big T as a phenotypes. So they are big. First law. Second law, the law of segregation. So in this one, we just talk about meiosis. So it means segregation means separate. So each of your germ cells only have one set of chromosomes. So therefore, you don't have two, you have one when they're doing this fertilization. So we already know that your first generation is big T, small t. Then your second generation is you use this first generation. So big T, small t, course was big T, small t. So then you got this F2 generation. So let's review what's this one, genotypes and phenotypes. So what's genotypes then? Combination of the alleles. So you can have three, big T, big T, small t, and the small t. So here you are. See this one? Big T, big T, small t. Then what's the ratio? Ratio of a genotype? Then it's 1, 2, and 1. And what's the phenotype? Here, phenotypes they show up. Expression, right? So you have dominant phenotype, which is tall. You also have the recessive phenotype is short. So therefore, you have tall. Who show tall? Big T, small t. So then you have 3 big one. You have only 1 small one. So the ratio is 3 and 1. Okay, remember these two ratios. 1 to 1, 3 and 1. Okay, done. So now let's go to the third law. Third law is saying that the law of independent assortment. So we write it down this one. So I show you one more time. Okay. So assume that you only have 4 chromosomes. Then you have 2 pairs. So let's assume that you have a gene located on these two chromosomes. So you have A gene, you have B genes. So you have 2 versions for each of them. So what you can see is with this one, if you inherited from father, both from fathers, then you get A, B, right? A, big A, and the big B. If you actually inherited all from your moms, then you get small a, small b, and so on. Okay, so that's 4 combinations, all right? No problem with this one. Okay, so now, look at this one. So let's say one germ cells is big Y, big R, another germ cells is big Y, small r, another germ cells is small y, big R, and the last one is small y, small r. So you have 4 different sperm. You also have 4 different eggs. So then you can cross this one, do Punnett square. Then you get a lot of different combinations. But the phenotype is 9331. Okay. So I think we have problem is the third one, because number one is really depend on how many pairs of chromosomes. So we want to give you 2 pairs. So how about 4 pairs? So you have 8 chromosomes, you have 4 pairs. You still can do manually for these different germ cells combination. So then you have 16 different combinations. You still can do manually, even in exam setting. But how about you have more? If you have 8 pairs, if you have 16 chromosomes, you have 8 pairs, can you still do manually? I don't think so. So you have to do mathematically. So what's the formula? Is 2. So each gene you have 2 alleles. N is how many pairs you have. So initially we talk about you have 2 pairs. So then let's do simple calculation. Order of calculation in biology is simple calculation. Okay. So you have 2 alleles, 2 pairs. Okay, you got 4, correct? We do this one. So the formula is working for you. So then let's go to this one. So you still have 2, correct? 2 alleles, but 4 pairs. So what you did do is 2, 2, 2, right? So you get 16. So that's also working for this one because you have 16 combination. So here you get 2 power 8, you have 8 pairs. So you get this much. So if you have 23, then you have this much different combination. Plus you have crossover. So then theoretically, none of humans individuals are the same. Okay, good. So let's review. What's the homozygous? Is big E or small e, right? So then what's heterozygous? Is big E, small e. Then the ratio of this one is 9:3:1. So you have D hybrid cross. Means that you have 2 pair of genes. You also have mono hybrid cross, means you only have 1 pair of genes. Okay. Now we're done. Okay. So use the mathematics method. Okay. So we actually got these 3 laws. They are fundamental. So now you have problem. I have problem with this because I cannot see the genotypes of this purple flower because the purple flower is dominant. So then you can have heterozygous dominant or you can have homozygous dominant too. So this is heterozygous. This is homozygous dominant. How can you see from outside? You cannot. You have to do experiment. What experiment you can do? This is called task force. So you can see what you do. So you actually cross this purple flower with the recessive phenotypes. So small p, small p. So then if this one is homozygous dominant, then they show all purple of the generation, of the children's generation. If this purple flowers is heterozygous, then you get half purple and half white. Okay. So this is course for task course. So what's the purpose? They want to find out if the purple flower is homozygous or heterozygous. Okay, so let's continue. Have you found that some weakness of further genetics experiment? Yes, I do, because the trees, come on, the trees grow bigger or smaller. It's not 100% depend on the genetics, because they also depend on soil, depend on whether your sunlight, whether you have enough water. So now, this another scientist used Drosophila to do genetic study. So what advantage of this Drosophila is, number one, they can produce a lot of offspring very quickly. And also they can generate next generation in 2 weeks time. And they only have 4 chromosomes. So you can actually grow them in the laboratory. Means that you control water, food, sunlight. So everyone will have the same. Good. So now let's do some calculation. So all the mathematics in biology is kind of precondition. So what's the precondition for this calculation for the Hardy-Weinberg equation? So this one is your population must be very big to meet the statistic value. Number two, the population must be isolated from another population, so you don't get this mixture because you don't want to change the allele. And also, you don't have mutants. Number three, the mating must be random. You don't have sexual selection. And you don't have environment pressure. That's why we use Drosophila instead of tree, because tree has a lot of environment pressure. So you have to meet these 5 criteria in order to do this Hardy-Weinberg equation. So now let's do it. What's this Hardy-Weinberg equation? So let's working on formula first. So formula is P plus Q equal to 1. So P is 1 allele, Q is another allele, so they can making a gene. So that's meaning of P plus Q equal to 1. So you can adopt this one to apply a square on both sides. So now you got P squared plus 2PQ plus Q squared also equal to 1. Okay, mathematics is simple, right? Good. So now let's try some examples. Okay, so let's do the first one. So question 1. Determine the percent of the population that hybrid 2PQ if frequency of allele in the recessive is 0.5. So let's back to this equation again. We have 2 equations. Okay, so what does this P, what does this P or Q mean? Okay, so let's see this one. So here, where P is the frequency of allele big A, dominant one. So when you talk about frequency, is talk about percentage, right? So remember, this is all talk about percentage, not actual number. So this is a percentage, okay? All of this is percentage. Got it? So percentage. So the Q is recessive of allele small a. So the recessive, okay? So again, percentage. Then these 2, you can treat them as they are in the germ cells because you only have 1 copy of alleles. So now P squared means that the percentage of individual has a big A. So they are homozygous dominant. Okay? So the Q square means that the frequency of individual have a small a, means that the both variants gave you a small a. Again, it's a percentage. So 2pq means that your body cells has a big A and also small a. Okay, now let's do this one. Okay, so they said recessive allele is 0.5. Then according to this equation, P plus Q equal to 1. Then you can see because the Q equal to 0.5, then P must equal to 0.52. Then what's 2pq then? Easy, right? So 2 multiplied by 0.5 and multiplied by 0.5 equal to 0.5. So this is answer. They ask you 2pq frequency. So let's go to another one. So here, determine the percent of population that homozygous dominant. They ask you big P, correct? And they ask you which one, big P or P squared. So you have to understand that. So P and the P squared are different. P just allele, and the P squared is talk about individual. They actually are body cells. So you can see the small p is germ cells, and the small p and the big P are germ cells, and big P square or Q square or 2pq are in your body cells. So now then say here, they ask you population that homozygous dominant. So they must have, come on, big P, P squared, right? So the P squared, they ask you P squared. So they already told you that the Q squared equal to 16%. So now let's do this one. See this one? So P squared equal to 36. So how can you get this number? Number one, you already been told Q squared equal to 16%. Then Q must be 0.4. Then your P must be 0.6. So P squared equal to 36%. So now it's your turn, okay? So now use this two equation, please. Do calculation. So remember, this is talk about percentage, okay? So we talk about percentage. Okay, we have problem with 19. So let's go through together. Okay, so now let's have a look. So you are correct. Question 9 is A. So you are correct for question 8 also. So let's do step by step. Okay, so that's no difficulties. Only if you do step by step. So let's go through the concept first. So your concept is P plus Q equal to 1. So in here, they already told you Q is equal to 0.4. So then P must be 0.6. Then 2PQ, heterozygous, is equal to 48%. So let's look at this one. So we already emphasized many times, say, all this equation is percentage. So here is not percentage. So this is actual number. You just convert it to percentage there, right? So you get 90. So 90 is blue eyes. You got 1,000 people. So 90 divided by 1,000. You got a percentage there. So this percentage is what? It's a P, Q, or P square or Q square. That's what you need to find out, right? So we already talk about it in here. We said here, the P and Q will not show any color or expression because they are in your germ cells. And your germ cells only have a single copy. The P square, Q square, 2PQ must be in your body cells because they can show some of expression. In this question, they actually say blue eyes. They're showing. So then they must be P square, Q square, or 2PQ. Correct? So then what you can see is here, right? So you actually say it's a Q square because there must be a smallest number and also they must be recessive. So then you got a Q square equal to 9%, or you can say P square. So then what you can see is the Q equal to 0.3, then P must be 0.7. Then heterozygous is a 2 times P, Q multiplied, then you got a 42%. So the tricky part for this one is they didn't give you a percentage. They gave you an actual number. So you have to convert this actual number to the percentage. Okay? So you have a lot of questions in your lecture notes. You can practice by yourself. Okay? So if you do have problem, we can talk about this one again. Okay? So I think we probably do this time. Do this question. Okay? Do this question. So the question asks you. Okay? So now. So remember, all the formula is percentage. If you actually have actual number, you have to convert that actual number to percentage. Okay, the first step is convert your number to percentage. So the concept for Rh blood, so you have ABO blood. Everyone knows ABO because ABO blood will write in your birth certificate. So you also have Rh blood. So Rh blood, Rh positive is dominant. So as long as you have one Rh antigen on your red blood cells, you are Rh positive. Rh negative are recessive. you must have two recessive, means you don't have antigen on your red blood cells, then you get Rh negative. Okay, so dominant and recessive. So let's try. So first step is convert the actual number to percentage. Okay, so let's go through together. So for biology calculations, they are required to go step by step. Okay, it's no point to jump because the mathematics is so simple. So most of the mistake is because you said, I know everything, I just use brainstorm to do the question. That's a wrong behavior, okay? So wrong manner. So I don't think this mathematics will cause problem because they are middle school maths. Okay, so now let's see this one. So let's review P plus Q equal to 1. So then you generate this second equation. So your question, right, as a Q squared equal to 16%. Why is Q squared not Q? Because they already told you it's RH negative. If it's Q, they never show any expression. So there must be Q squared. So that's first step, the concept. So then if you get this right, then you get a Q equal to 0.4. Then the P must be 0.6. That's it. So simple, right? So they ask you, how many students would you expect for each of the possible genotypes? They ask genotypes here. So the genotypes is combination of two alleles, correct? So therefore, AA is one. Then big A small a is another one. And the small a is the last one. So that's genotypes. So therefore, you get this one. So P squared multiplied by 2000, you get 720. And 2PQ percentage multiplied by 2000, you get 960. And recessive, so the Q squared multiplied by 2000, then you get 320. So the question asks you is genotypes. So genotype has two alleles. Okay, so then your answer here is big A, 720, 36%. Big A, small a is 960, 48%. Small a is 320, 16%. Done. Okay. So if you're still confused, think about this concept. Okay. And do step by step. Okay. So now let's go on. So if we have a genetic problem, or even you said, oh, look, we have not really a problem, is a genetic inherited. So we want to retrieve family history, such like certain expression. So you can draw this called a pedigree tree. They will allow you to get the idea how about your family history of this genetic symptoms. Okay, so in general, the circle is represent female, square is represent man. So this solid one is symbolized as diseased or symptomized. So the empty one is no disease, no symptom. Okay, so now you have those are two kind of disease. One is called autosomic disease. Another one is a sexual linked disease. So let's talk about those two, okay? So what's autosomic? So autosomic 12 means that you have the gene located on number 1 to number 22. Okay. So for instance, here you have a gene located on number 2. Okay, this is belong to autosomic. If you have disease, they call it autosomic disease. So you also have a dominant one or you have recessive one. Okay, so now let's talk about sexual linked recessive disease or sexual linked dominant disease. So number one, your gene must be on X or Y chromosomes. So you only have one or two of this sexual chromosomes. If you're a girl, you have double X. If you are a boy, you have X and Y. So then this is 2. So then you can have recessive, you also can have dominant. So now We already done a lot of this autosomic, right? All this before we talk about autosomic. So now let's talk about sectioning. So this is your sectioning. So sectioning, let's start with X, because X is most common. And so sex-linked disease is most commonly happened in X chromosomes. Why? Because X chromosomes are much bigger. X chromosomes are much bigger than Y. They contain more genes. That's number one. Number two, X-linked disease most commonly found is recessive disease. So let's look at those recessive disease on X. So number one, muscular dystrophy. I saw this person on the street in the mall. Okay, so they actually cannot stand up when they're 20. It's very poor, okay, because they have muscle wasting. Another one is the highlights, especially for men. So sometimes you walk on the street, you see, oh, this young man, maybe 30 years old age, they don't have hair. So this is kind of disease. So they call it borderless. So another one is colorblind. So someone cannot see difference between red and green. So last one is quite severe disease called hemophilia. Hemophilia is very common in African countries. So when you cut your fingers, they cannot stop. You have to go to hospital. Okay, so let's look at this one. Most mild disease is borderless. Okay, so now let's see. So here is on X chromosomes. They are recessive. So if the girl has the one, they don't have symptoms. They didn't show up. But unfortunately, if man has one, then they got disease because X, Y carry different genes. They cannot help each other or they cannot dominate each other. Okay, so now let's assume that the father is okay, no problem, absolutely normal. But mom is carrier. So what do you mean by carrier? Means that they carry a disease genes, but are not showing symptoms. Concept, okay? Carry disease genes, but not showing symptoms. It's called carrier. So now you can see they have two girls. So the two girls is one is absolutely normal. Another one is a carrier again. So both will not show any symptoms. They appear normal. So this one, the boys, you can see one is absolutely normal and the one is showing disease because they got an X from the mom. The disease X from the mom. Okay, so you can see if mother's carrier, then you can see the boys get disease, the half boys get disease, and the girl hasn't got disease. So now we can draw a conclusion that the X-linked recessive disorders predominantly affect men more. Okay, so one thing you have to know that any girl or boy, you only can have one mom and one dad biologically. Okay, you cannot combine. Okay, so now let's try this question. Okay, do you remember to do pentagram? quite panic, right? So let's go through together. Okay, so let's go through this one. So number one, the man's disease. So father's disease. So this means that the father has XD disease on X. So they show symptoms. So then it's the mother. Mother is no symptoms, appear normal. So there can be one is absolutely normal. So mom's absolutely normal, no carrier. So then when you do Punnett squares, you get all of girls, all of daughters are carrier. All of the sons are normal. So which one? E, correct? So this one is for E. Okay, no problem, right? So this is for E. So this situation, if mother is 100% normal, then your answer should be E, correct? So let's talk about the second situation. The second situation is mom's carrier, but not showing any symptoms. So then you can see daughter, Y is carrier, not showing symptoms, appear normal. Then second daughter get disease. Then son is Y is normal, another one is disease. Okay, so now can we find this one in these 5 choices? Answer is no. So let's see D. Someone, I got 2 or 3 students answer D. They said approximately 50% of the son will have X-linked disease. Good for this one. So not approximately, should be clean cut. 50%, 25%. Right? So for this one, I assume that, okay, I give you credit for half of the son get disease. Then this one, and the order of the daughter will be carrier. So the daughter, Y is carrier. So I already gave you a definition. What do you mean by carrier? The carrier means that they carry disease genes, they carry disease alleles, but not showing symptoms. That's called carrier. But they have one carrier, but one is disease. So then this one is wrong because not all of daughter are carrier. Okay, so your answer is E. Okay, so now let's talk about another one. So I leave this one to do by yourself. So let's talk about Y-linked trials. So number one, Y-linked trials are very rare. And number two, the Y-linked trials are always dominant because the boy only have one Y. The girl don't have Y. So the X cannot cover Y. Okay, good. So another thing we want to talk about is called X inactivation, foreign body. What's foreign body? So in the mammals, for the female, you have 2 X chromosomes, but one is not working. One is take a break. They call it inactive. So such like cat. So one X is inactive. Okay, so if you have a 2x, they only happen in the female because only female has a 2x. So if it's boy, you don't have this choice because you only have 1x. So now let's talk about this one. So your fur color is on X, is controlled by X genes. So now what you can see is here for the female. Okay, so you have two versions. You have a black version, you also have orange version. So the black one will actually is showing black, and orange one will showing orange. They both dominant. Okay, so the black dominant and orange also dominant. So now if you both X code for black genes, okay, you have a black cat. If both X carry orange genes, okay, you show orange. So the complicated occur is that here, so you have one orange and one black. So then they will take one is inactive. So for instance, here, so black X is working and the orange is not working. So what you can see here is that, look at this one. So you can see they will show black because your orange is not working here. Then if you get another area, the orange is working, the black is taking rest. So then you get orange. So this indicated on here. So the black area means that the black is active. And the orange area is orange active. So this only happened in the female, right? Because you have two. You have two. It wouldn't happen in the male because they only have one choice. So you can have orange, you show orange. If you have black, you show black. Okay, good. So now let's do this question. Good, I got one answer. What do you think? So let's see that we must say here. So let's go through together. So the man cannot be a Kotka cat because they only have orange or black. They don't have mixed color. So man, so number one, of course. Right? So let's see this one. So here, the female can making cortical male. Okay, so took a male down. So we only have those two, correct? So let's see this one. The first one, a black female, and the orange male can produce a black male. Yes, of course. Yes, yes. Right. As long as they have black eggs, there will be black. Okay. So what's your answer then? So this one should be okay, right? So this one is okay, right? So you get hard for this one, right? Okay, so another hard for this one. So this one is okay, right? This one's okay, right? This one, last one is wrong because you cannot have cortical cat. Simple as that. Okay, so your answer is D. Okay, we finished today. Okay, so if you have any concern, just email me. Okay, please. Thank you. Bye.